Assalamu alaikum class. Today we're going to study about antibodies, that is their structure and function. So the learning objectives of today's lecture are to understand the characteristics of antibodies, their basic structure, to define isotypes, allotypes and idiotypes, and to understand the different classes of antibodies, their structural and functional differences, and how these antibodies provide protection against infection. So let us start today's class. An antibody is also known as an immunoglobulin. It is a protein that is produced by the plasma cell after stimulation by an antigen. So antibodies are always produced in response to an antigen. And these antibodies can recognize and bind to the specific antigen against which they are produced. Depending upon the electrophoretic migration, three types of globulins are present in the blood, namely alpha, beta, and gamma. Antibodies belong to the gamma globulin class. Antibodies are synthesized by the B lymphocytes or B cells and are secreted by the plasma cells. So these antibodies can be present in the blood, in the tissue fluid, in the gastric and mucous secretion, in breast milk. So they are used by the immune system to identify and neutralize pathogens such as bacteria and viruses. And they also mark the microbes for destruction by phagocytosis before they can infect any cell. Antibodies can be present in two forms in our body. Generally, antibodies are in form of secretory antibodies. Like here you will see, they are secreted by the plasma cell and they move freely in the blood of tissue fluid. And sometimes they can also occur in form of a binded form, like in case of B cell receptor or BCR. So these BCRs are actually antibodies which are bounded to the surface of B cells. Now let us discuss the characteristics of antibodies, that is their specificity and their biological activity. Uh, let's discuss specificity first. So by specificity we mean that the antibody has the ability to bind to a specific epitope. So one B cell will produce antibodies that will bind to only one epitope. So one B cell will produce antibodies specific to an epitope. However, all the clones of this B cell will have the same specificity. The second characteristic is the biological activity, which means how the antibody is able to protect the system from the pathogen. So the antibodies can trigger protective physiological activity generally after binding to the antigen. So antibodies can facilitate phagocytosis by opsonization of microbe. They can cause activation of complement system, which results in clearance of antigen. They can trigger an allergic response, and most importantly, they can reduce the virulence of microbes by neutralizing them and their toxins. Now let us study the structure of antibodies. All antibodies share a basic structure, that is a Y-shaped tetrameric structure as shown here, which is held by these disulfide bonds. So this Y-shaped tetrameric structure is composed of four polypeptide chains. The green ones shown here are called the light chains, which are composed of 220 amino acids, and these two are identical to each other. And the larger chains or the heavy chains are the ones which are composed of approximately 440 amino acids, also identical to each other. There are two basic regions which are of biological importance, called the binding site or the fragment of antigen binding called FAB or Fab region. This is the region which determines the specificity of the antibodies. This region is also called apparatope. Actually, this is the region which binds to the epitope and recognizes it. So this region is composed of both light chain and heavy chain regions. So an antibody, as in this case, is bivalent because it has two sites of attachment for epitopes. The second region, which is of importance, is called FC region or fragment of crystallization. It's basically composed of only heavy chain, and it is the region which binds to the complement system or NK cells. Now let's study the structure of antibody in detail. An antibody molecule is composed of four polypeptides, two identical heavy chains that are connected to each other to form the Y-shaped formation, and two light chains. Disulfide bonds between the cysteine amino acids of these molecules or polypeptide connect each other. So there is one disulfide bond between the light chain and the heavy chain, and the disulfide bond between the two heavy chain can range from 1 to 13 depending upon the type of antibodies. One end of the antibody chain is called N-terminus, the other end is called C-terminus. So as you can see, each chain is composed of a V stands for variable region and C constant region. So a light chain is composed of two regions, one variable and one constant. So VL stands for variable region for light chain and CL stands for constant region for light chain. Similarly, the heavy chain also has variable region and constant region. So they also have one variable region. However, the constant region can be 3 or 4 depending upon the type of antibodies. So this is the variable region which determines the antigen binding specificity of the antibody. So a variable region is composed of a light chain and a heavy chain region. And the FC region, as you can see here, is composed of only heavy chain molecules and it is towards the C terminus. And this is involved in complement system activation and NK cell dependent cytotoxicity. One important thing you can note here is that these variable regions are two in each antibodies. So they are composed of two fragments of antigen binding. So we notate this as FAB square.